Oh, hello, I'm Pancho Besanilla from the University of Chicago, and in this video we would like to summarize highlights from our recent paper in Neuron. On this paper we use cell-targeted gold nanoparticles that enable inactivation of neurons by light.
Hello, I'm David Pepperberg from the University of Illinois, Chicago. In this video, we'll first present an animation that describes the technique and the mechanism of photoactivation suggested by our data. We'll then illustrate some of the electrophysiological and imaging results obtained, and then conclude with a comment on the technique's possible future applications.
Here, we will demonstrate our technique for using gold nanoparticles to optically stimulate specific neurons without needing genetic modification. First, we will review the necessary components for this technique to work. There are several features of the target neuron that play an important role in this method. First, the basis of this technique is to change the membrane capacitance of the target neuron. We also need a surface antigen to use as an anchor for the gold nanoparticles. This can be virtually any membrane protein. Finally, the voltage-gated sodium and potassium channels will allow the cell to respond to the stimulus by firing an action potential. To produce optical stimulation, we need to add two additional components, both of which are commercially available and can be used with no further modification. First, we need a primary antibody to target the chosen surface antigen. The choice of antibody will determine the targeting specificity of the nanoparticles. Second, we need gold nanoparticles which are ligated to secondary antibodies appropriate for the chosen primary antibody. We begin showing a native neuron at resting potential. To use this technique, we first deliver the primary antibodies, which bind to their target antigens, and then we wash out unbound antibodies. Under this condition, a pulse of green light has no effect on the neuron's membrane potential, as shown by the flat trace to the upper right. We next add gold nanoparticles conjugated to secondary antibodies, followed by washout of unbound nanoparticles. This yields nanoparticles anchored to the membrane antigen through an antibody linkage. Now, a brief pulse of green light absorbed by the nanoparticle is transduced into a brief localized pulse of heat that causes a transient increase in membrane capacitance. This leads, in turn, to cell depolarization. If threshold potential is reached, voltage-gated channels open and an action potential is triggered. These stimuli can be repeated at rates of at least 40 Hz with perfect fidelity of neuron firing. Importantly, stimulation depends on the rate of change of temperature, not on temperature itself. In fact, we measure an increase of less than 2 degrees total in temperature near the neuron. Although here we show antibodies as the linking molecules to target specific neurons, our paper also demonstrates that a scorpion toxin with affinity for voltage-gated sodium channels can serve this role. This suggests that a wide variety of anchoring molecules could potentially be used to attach gold nanoparticles to a targeted subset of neurons or other excitable cells.
Hello, I'm Bobo Dan.
TSE, the toxin binds to the voltage-gated sodium channels with high affinity without blocking them. In this work, we chemically synthesized the TS1 toxin and incorporated an alkyne group onto TS1 without compromising its biological activity. We then used a click reaction to attach one biotin to each TS1. Biotinolated TS1 was then conjugated to streptavidin functionalized gold nanoparticles for electrophysiology studies.
Hello, I'm João Carvalho de Souza.
As Bobo mentioned, we have used TS1 to attach gold nanoparticles to neuronal membranes. Before adding the TS1 functionalized gold nanoparticles, no voltage change develops when a 1 millisecond pulse of light is presented. However, when we attach gold nanoparticles to the neurons, an action potential is triggered by light. Furthermore, the temperature measured close to the neuron increases by less than 2 degrees during the light pulse. This technique enables stimulation of transaction potentials at up to 40 Hz in DRG neurons, and even this frequency is limited not by the technique, but instead by intrinsic properties of the neurons.
Hi, I'm Jeremy Trigger.
To test whether this technique could function in a complex neural tissue, we injected TS1-labeled gold nanoparticles into slices of mouse hippocampus. We then imaged the site of injection with an infrared voltage-sensitive dye to monitor activity in response to pulses of green light. We found that optical stimuli led to transient depolarizations in the injection region. These depolarizations resolved back to baseline levels of activity over the course of several hundred milliseconds. This time course could be spatially mapped, showing the pattern of depolarization and hyperpolarization at different time points following the stimulus.
The technique we've introduced here may be broadly useful in research on excitable tissues. It complements the technique of optogenetics and could serve to screen targets for later optogenetic treatment. Ultimately, the present approach might also have therapeutic applications. For example, in certain eye diseases where the retina's light-absorbing photoreceptor cells are no longer functional, a degree of vision restoration might be possible by treating remaining healthy cells in the retina to make them light-sensitive. Much further research must be done prior to any such clinical application, but the present results encourage research to explore this possibility.